back to lipids. What word, when you think of lipids, you have lipids in your diet, but what would we call them? Fats. What are other examples of lipids? Oils.
Waxes.
Fats, oils, waxes. All of which are things that aren't going to dissolve in water. They're going to repel water. Waxes provide waterproof coverings for organisms. Whether it's like plants, you've seen the plants with the leaves with really shiny coatings on them. That's because they have a wax coating on it to help prevent water loss. Ducks produce oils that help keep their feathers from absorbing water and keeps them not waterproof, but water repellent. So that's what we're looking at, and there's no way we're going to look at all of the polymers of lipids. We're just going to talk about a few. And when we defined a monomer, we said it was like a chain of repeating monomers, or polymer was a chain of repeating monomers, right? Well, lipids are kind of different where it's not a chain of repeating things over and over, but it is a big molecule made of little pieces that come together to make it. So our monomers are fatty acids, and there's two types of fatty acids. Anybody know how they're classified, Brian?
First one, saturated fatty acids.
Okay. How do you know it's saturated?
Because it doesn't bend.
Okay, so it's straight. So that one's saturated. What is this one? And it's bent, right? Now, if you were to jump in the lake with all your clothes, shoes, everything, you get out and say, man, I am saturated. What do you mean? You're wet. All of your clothes, shoes, everything have absorbed all the water they can absorb. That's what saturated would mean in that situation. Why do we call a saturated fatty acid saturated? Well, look at the picture compared to unsaturated. It doesn't necessarily have to do with water. I'm saying that's how we would use it if we jumped in the lake. Meaning we'd absorb all the water. There's no more room for water. Okay, it does tend to be solid. But why do we call it saturated? Look at the pictures. Good. Look at the saturated compared to the unsaturated. Are there spaces where you can add more hydrogens in the unsaturated? But what about the saturated? Is there any more room for any more hydrogens? So it's saturated with hydrogens. When we get to the unsaturated, there's some hydrogens missing. And where it bends, what's present between the carbons? Double bonds. Now, if you look at this fatty acid, it's mostly carbons and hydrogens, right? Does it fit the ratio of a lot, a little? A lot of carbons, a lot of hydrogens, a little bit of oxygen. How much oxygens are on these fatty acids? 2, right? There's a carbon, double bonded to an oxygen, and single bonded to a hydroxyl. There's a COOH. What functional group is at the end of the fatty acids? Carboxyl. So a fatty acid is a I can't read that, can you? A fatty acid is a carboxyl plus what we call a hydrocarbon chain. What do I mean by hydrocarbon? Hydrogens and carbons. So we have a carboxyl group and hydrogens and carbons. Now, fatty acids can store lots and lots and lots of energy. Because every carbon-hydrogen bond is a high-energy bond, meaning it can store lots of energy in it. So what function do you think fatty acids might have? as a molecule, what's its function going to be? To store energy. Do you think if carbon-hydrogen bonds are high energy bonds, what would have more energy stored in it, a fatty acid or a glucose molecule? Fatty acid, because it has more carbon-hydrogen bonds, so it has more energy stored in it. And in fact, we talk about energy, and read in the book that energy can be measured in calories. The amount of amount of energy that it takes to raise 1 gram of water 1 degree Celsius. Do you remember reading that? Well, where do we see the word calories? On the boxes of food we eat, right? When they're talking about calories, they're saying how much energy is stored in that food. Well, if you look at the calories from fat versus how many calories you get from the carbohydrates, you get more calories per gram from fat. You get about 9 calories per gram from fat, and you get about 4 calories per gram for carbohydrates. So a lot less energy. And in 2 units, we'll be setting food on fire to see how much energy is in it. It's always a fun lab. It's a stinky lab too. But we have 2 fatty acids, saturated, unsaturated. Those are 2 of our monomers, 2 of our building blocks. The other building block is called a glycerol. A glycerol is a 3-carbon chain, and each carbon has a functional group attached to it. What is that functional group? Hydroxyl. So it's a 3-carbon chain with a hydroxyl attached to it, to each carbon. Now, we can put this glycerol together with fatty acids and get a polymer. When the fatty acids attach to the glycerol, what process is going to be used to attach a fatty acid to a glycerol? Dehydration, synthesis. And when it attaches, it's going to form a covalent bond. What is the name of that covalent bond? The ester linkage. So here we have a glycerol, and we have 3 fatty acids. What do you think the name of that is? Anybody know? Tri for the 3 fatty acids, and glyceride because they're attached to a glycerol. So it's a glycerol plus 3 fatty acids. What do you think its function is? And it's long-term energy storage. It's not like our carbohydrates. Carbohydrates are going to store energy. Monosaccharides are quick energy. Starch and glycogen, they're more short-term. They're the first source of energy for animals is to go to glycogen. This is more long-term. This is why, like, if people don't eat for a while, they're like, oh, I gotta take my prom dress, so I'm not gonna eat. I'm gonna eat lettuce every day, right? And so they don't eat for a while and they lose weight. Well, then they start eating again after prom, and the body's like, I don't know where I'm gonna get I don't know when I'm gonna get another meal, so it takes everything and it stores it. But it's not gonna store it as carbohydrates. It's gonna store it as fat. Because fat takes a longer time to break down. It stores more energy for longer periods of time. So that's why we see people, like, they'll go on these crash diets where they don't eat a whole lot and they lose weight, and then they gain weight and they gain more than they actually had on them to start off. Because their body is storing it very quickly and it's storing it as fat. So that it has storage for later. It has energy for later, the next time they decide to not eat. Alright, so this is one polymer, triglyceride, long-term energy storage. Next polymer, what is this one called?
Phospholipid.
Phospholipid. And phospholipid is made up of a phosphate group, a glycerol, and 2 fatty acids. And those fatty acids can be saturated or unsaturated. Now we call the head, that where that phosphate group is, we say it is hydrophilic, which means it loves water, right? So if it's hydrophilic, is it polar or nonpolar? Polar. And we call the tails hydrophobic. If they're hydrophobic, are they polar or nonpolar? Nonpolar. What is the term we use for a phospholipid? It's both hydrophilic and hydrophobic. Now remember it? It has both personalities. It's amphipathic. Amphi means both, pathic means personalities. It is amphipathic. It is both hydrophobic and hydrophilic, both polar and nonpolar. Where do phospholipids function? Where do we find them? In the membrane bilayer, right? Then the last one?
Cholesterol.
It is cholesterol. Can also be classified as a steroid. Its biggest characteristic are these 4 carbon rings. It makes it very easily identifiable. When you see these 4 carbon rings, you should think cholesterol or steroid. You could also think hormone, because cholesterol is the precursor to a lot of hormones. Name a hormone. Estrogen, testosterone. Cholesterol is what is used to make those. And hormones are chemical messengers. They carry messages throughout the body, telling the body to respond certain ways. So, but those are lipid hormones. Some hormones are proteins, but a lot of them are based from cholesterol. They're lipid proteins. Questions? All right, so your job now is to answer all those questions under fun facts and critical thinking. You could discuss it with the people around you. If you don't have a partner, turn to someone near you and discuss it with them. I'll give you a few minutes to try your best.
Alright, so, I'd say they I didn't know. You start to work one phosphate group, draw like
I always say
it's like imagery of that paper.
Just a bunch of them.
It's this one, right? No, I know
All right, we're gonna do these a little out of order. Y'all okay if we start, or you need another minute or two? Or another minute or two? Okay. All right, let's start with drawing a phospholipid bilayer. Chloe, how am I gonna draw it? Circles on top.
Yeah, and then instead of going in the leaves, I can see the one on the bottom.
Y'all agree? Why do we draw like this?
Is that how you, if I did really, each other?
Okay, so my hydrophobic tails, they interact, they're attracted to each other, but why do they go to the middle? They don't want water, and where's water? There's water here, but where else is there water? On the inside. So now up here it says you have a container of water, and you drop phospholipids in it. How are they going to arrange themselves? Okay, so maybe it arranges it with a bilayer and it traps water in the middle. It could. What else could happen? What? Could it do this? It could do that. But now, let's say I have a container, and it is filled with water, airtight, and I drop the phospholipids in it. Maybe they form a bilayer with a double circle. Could they do this, though? Because those tails repel water, right? Those tails go in the middle, and we have this circle where all the heads are facing the water, and there's no water on the inside. Could it do that? But how's it going to keep the water out? I mean, I think it could. It could. But why would it do this instead of this? What's the difference? Why do we have a bilayer here? Why would I not have a bilayer there? What couldn't be on the inside here? Water. Are you with me? So this is called a mycel. And if there was a drop of oil in here, oil is a fat or a lipid, right? You would see those tails congregate around that because those tails will be attracted to that lipid and we form that mycel. You ever read micellar water or seen it? It's used to like clean faces. Well, what is all over our face? Oil. So when you clean your face, you're taking oil off, right? Well, they have phospholipids in that water, and it actually goes and surrounds the oil and pulls it off your face. That's the idea behind it. If you buy it, whether it actually works that way, that's actually what they say it is a different story. But that's the idea behind it. Questions? Okay. Okay. What are the bonds between glycerol and fatty acids called? An ester linkage. And all polymers are formed through a process of what? Why do I keep asking this question? Because I don't want you to forget it. And is it common between all of these macromolecules? All right, cholesterol is used by the body to make hormones such as what? Estrogen and testosterone. Hormones do what? Oh, a lot of, I don't know what you said. Okay, they can help regulate functions because they do what? They carry chemical messages. And they carry them throughout the body. And why are lipids more efficient energy storage than carbohydrates? Right, so we did CHO and CHO, so now we gotta do what? Sean, what are we doing next? Proteins.
Oh, gosh.
Proteins, what elements are in them?
Carbon, hydrogen, oxygen, nitrogen.
And what functional groups do we find in proteins?
Enzyme.
Okay, enzymes are a protein, but remember the functional groups are the groups of atoms that we talked about yesterday. We talked about 4 of them, and there are 2 that are present in every single amino acid, and they're present in protein. So they're present in protein. What are they? And what else? And remember, your amino group is NH2. You can also see it as NH3, the plus sign. And carboxyl is your COOH, or it looks like this.
You want to write that?
You don't have to, I'm just reminding you what it looks like. What is the special name between the monomers of proteins? You already know this one. Okay, well, polypeptide is multiple of them, so what is the bond called? Peptide bond. So, monomers of proteins, what are they called? Amino acids. And how many different amino acids exist? 20. 20 total different amino acids to make up all the different proteins in the entire every living organism everywhere. From bacteria to us, all the proteins are only made out of those 20 amino acids. So what makes them, the proteins of bacteria, different from the proteins in us? Okay, well, the structure or the shape of them, but what makes, they're all made out of the same 20 amino acids. They have the same amino acid pool to choose from. Okay, the sequence of the amino acids, the way they're arranged, changes the protein. But every amino acid has a general structure. Every amino acid has these things. It has this right here, NH2, what is that? The amino group. It has a carboxyl group. It also has this carbon in the middle. It's called the central carbon. And it has what's called an R group. And R groups are what make the amino acids different. The R group is just referring to it's a group of atoms that when it attaches to it, it gives that amino acid certain properties. So how many different R groups are there? 20 different amino acids. And this is the only thing that changes between them, so there has to be 20 different R groups. So R groups, sometimes they get called side chains. Are what determine if an amino acid is polar or nonpolar, charged, hydrophilic, hydrophobic. In order to take two amino acids and put them together to make that peptide bond, what process has to occur? Dehydration synthesis. Now, amino acids, if you notice, there's an N, a C, and a C, right? You see that? Amino acids have a directionality. They can't ever combine where we have one amino acid here and the other, oh, I keep writing it backwards. The other one is like that. They only combine like this. Where the nitrogen of one amino acid, or the amino group from one amino acid connects to the carboxyl group of the other. So, if you're looking at a structure and you see an N, and a C, and a C, and an N, and a C, and a C, what would you know about that structure? It's a bunch of amino acids, so it must be a protein. What if you just saw just this right here, and you just saw NCC? What would you know it is? It's an amino acid, because it's just one of them. So that NCC pattern is a telltale sign of telling you that the protein, amino acids, you look for that NCC pattern. Does that make sense? Question. Feel okay? Yeah. What determines the order of the amino acids in a protein? The what?
DNA.
The DNA has the code, and then it gives the code to what? mRNA, and then mRNA, we read the code on. How many bases is the code on? 3. So, here's what I want you to look at. These amino acids that we're looking at, this is the picture from your textbook. These are the 20 amino acids that exist. See the chemical structures? Here are some names. Glycine, GLY, is an abbreviation. Alanine, ALA, is an abbreviation. Serine, SER, is an abbreviation. Do those sound familiar? Do you remember using those on the little chart where you found the codons and you figured out what amino acid it coded for? This is what it was coding for with these chemical structures. How many of you think you can memorize this? By tomorrow.
Oh, no. That's funny.
Auto test.
Are we getting matching?
I'm going to give you the pictures and you're going to tell me the name and its properties. Hydrophilic, hydrophobic, polar, nonpolar.
How many points?
What?
How many points? I
5,000.
thought you were actually saying there was a thing.
I am.
Oh, there really is?
But does it matter how many points? Are you only going to study if it's 5,000 points?
No, I'm saying that's like a lot to remember in one night.
On top of what we're reading, on top of.
Yep.
Other classes.
Welcome to AP.
Welcome to AP, man.
I'm telling you, I'm not going to college.
Yes.
School sent it to me.
When I was in AP biology, I had to do that. You don't.
Oh, thank God.
You have to recognize NCC and tell me it's an amino acid. Okay? But when you're reading tonight and you're really mad at me and you're studying, just think it could be worse. It's not that bad. See, I just made myself look good. All right. See the purple boxes? All the purple boxes are exactly the same. That's that general structure, the NCC pattern. The colored boxes, the yellows, the greens, the pinks, the blues, what are they? Those are the R groups. That's what gives them the properties. If you notice down here, these have a charge to them. If they have a charge, a positive or a negative, could they be attracted to water? Yeah, because water has opposite poles. So if I saw these and I saw that charge on there, could I look at it and go, oh, it's going to be hydrophilic? Yeah? All right. Look up here at these. They're mostly carbons and hydrogens. What is that similar to? Lipids. Are lipids polar or nonpolar? Are they hydrophilic or hydrophobic? Hydrophobic. So when the R groups are mostly carbons and hydrogens, those amino acids are probably what? Hydrophobic and hydrophilic. Hydrophobic and nonpolar. You don't have to memorize them, but you can take things you know and look at them and figure some things out as well. All right, here's what I need you to do. I need you to find a piece of junk paper. You're about to tear it. Find a junk paper. Something you don't want anymore. Tear a strip lengthwise.
How tiny. Does it matter?
Get there to write on.
Oh, that'd be blank.
It doesn't matter.
I'm gonna cut the strip.
You're going to throw it away. So,
Oh, it don't matter.
tear a strip, and on that strip, I want you to write 10 abbreviations for amino acids. If you can't see them, you can walk up here and see them. But if I want you on your strip of paper, this is my strip. Write the abbreviations like this. But you're gonna write 10 of them. If you can't see them, come stand up here and look.
Like big or small?
Oh. Going like this. Right in across. It doesn't, however big you want to write it, just write your 10 amino acids. You can use the same one more than once. You can, whatever order, but write them going across the paper like this. Write 10, as quick as you can. If you can't see, here's some abbreviations. Tyrosine is TYR. Asparagine is ASN. Serine is SCR. Methionine is MET. Aspartic acid is ASP. Once you have 10, you can stop. You can use any combination. Does every protein have a specific function? What gives it its specific function? Structure, shape, right? What determines its shape? The sequence of amino acids, right? So in order for a protein to get its shape, it has to go through several steps. And you're about to learn about those steps. So You have your list of amino acids. And we're going to talk about these different steps. The first step is called the primary structure. It can be abbreviated with 1 and a degree sign. That means primary. So the primary structure. To make a protein, what's the first thing you have to do? What do you have to put together to make a protein? Amino acids. So your first step is just simply a chain of amino acids. And the sequence of amino acids is determined by the DNA and the RNA that code for it. So when I link a bunch of amino acids, what type of bonds are going to form between those amino acids? Peptide bonds. So on your paper, draw peptide bonds between your amino acids. Just draw a line between each one of them. So I just literally have lines between them. I form peptide bonds. Now look at your neighbor's sequence. Does your sequence match your neighbor's? Elena and Sammy show each other yours. They don't match, right? Because your DNA made yours and their DNA made theirs. Alright, so they don't necessarily match. This is your primary structure. It's just a chain of amino acids. Does it have a real specific shape? Is the shape of yours pretty similar to the shape of your neighbor's? Yes. So the first step doesn't really give it its shape. But the next thing that happens starts to give it its shape. It forms what's called secondary structure. And depending on the protein and the amino acids present, it can either form what's called an alpha, an alpha helix, or a beta pleated sheet. So it could do this. Take your paper and kind of twist it and make a helix out of it. Is it getting kind of a different shape?
Yeah, bro.
So it could do this, but because it's paper, this one's kind of hard for us to simulate. So this is one thing it could do. The other thing it can do is it can form a pleated sheet. What is a pleat?
Like this.
Right. It's a fold. Right? So you're going to pleat yours the way you want to. Do they all have to be the same? No. Do they have to be equal folds? No. You pleat it however you want, because what's going to determine how it pleats? The sequence of amino acids. So this is my beta pleated sheet, my secondary structure. Hold yours up and look at the other people's around the room. Do they all look the same? Some of them have small folds, some of them have big folds, some of them have 10 folds, some of them have 3 folds. Right? They're different, but is it starting to get a shape? Now, would it be kind of easy for it to straighten back out again?
Yes.
Yeah. So there has to be something to kind of help it keep this shape. Well, there are slight interactions between some of the R groups. And it's a very weak attraction or bond. What do you think it might be? Hydrogen bond.
Ooh.
The next step is where the protein gets its actual functioning shape, and it is called the tertiary structure.
Why are they measuring the rooms?
That's just a symbol to mean like primary and secondary. It's not a measurement in that regard. This is when it gets its shape, and it's basically folding up on itself. So has anybody ever taken like a lanyard or something and you start twisting it? And the more you twist it, what happens?
Like on the swings.
It gets tighter. I have loose circulation in my fingers. But as you twist it, does it start to kind of fold up on itself? And now it's gotten a new shape? That's what's happening. This would be what happens with the alpha helix. The beta pleated sheet, it's going to fold up on itself as well. So now I want you to take your beta pleated sheet and I want you to fold it however you want. You can make it look like a swan, you can make it look like a swan. Whatever you want to do, but you fold it. And you make it have the shape you want it to have, but do it quickly. I had an origami artist in the last block. We had ducks.
I think mine was pretty good.
When you have your tertiary structure, hold it up. And see if yours matches the shape of anybody else's. So this tertiary structure, the way it folds up is determined by the order of those side chains or the R groups and the way they interact. Is a polar R group and a non-polar R group, are they going to be near each other? Think about it. Can we put something non-polar in water and they want to stay by each other? No. So can we put a polar R group and a non-polar R group by each other? No, they would repel. So we start to see all of our non-polar and our hydrophobic amino acids, they fold to where they're near each other, and we see what are called hydrophobic interactions. We also can see a few ionic bonds form between our groups. We can also see another kind of bond form. So if you look here, there's an amino acid, and here's the R group, and here's an R group for another amino acid, and they both have a sulfur. When two sulfurs from the R groups are near each other, they form a bond that's called a disulfide bridge. A disulfide bridge. So those are the bonds that are holding that tertiary structure in the shape that you just folded it in. And sometimes that is a functional protein all by itself. But some proteins are more complex, and they have to have a quaternary structure. The quaternary structure is where multiple tertiary structures come and fit together to make the fully functional protein. And those are held together by hydrophobic interactions. And if that doesn't make sense to you, think about being in a haunted house, and there's 4 of you, and you're walking through the haunted house, and all of a sudden, a bunch of clowns jump out, and you're all afraid of clowns. What do? You jump toward each other, you link arms, you stay together, right? So these tertiary structures, the areas that are afraid of water, they link together. They are attracted to each other and stay together to make that functional protein. So this shows you the quaternary structure of hemoglobin. It's made out of 4 tertiary structures. that work together to transport what molecule? What does hemoglobin transport? Oxygen. Right, so it's in our red blood cells to carry oxygen. So that's how a protein gets its shape. What if we were to put it in something that caused all the hydrogen bonds to disrupt? There's no more hydrogen bonds in a protein. What structure is that protein left at?
Just straight line.
Which structure?
Primary.
The primary structure. If we lose the hydrogen bonds, can the secondary structure be there? There's no secondary structure. Is there a tertiary or quaternary? So all we're left is with the primary structure and the peptide bonds. Anybody got questions? Did folding the paper help a little bit? Maybe? At least woke you up if it didn't help. Or partially woke you up. Functions of enzymes. Enzymes some functions of proteins. They are the most varied group of macromolecules we have. There is nothing that happens in your body that doesn't involve proteins in some way, shape, or form. Proteins can be enzymes, like our digestive enzymes. What are enzymes? What's the C word we use to describe an enzyme?
Catalyst.
A catalyst, which means it speeds up reactions. They can be used for storage. They can be used for communication. What does insulin communicate? What does it tell the body to do? What does insulin tell the body to do?
The liver.
Tells the body the blood glucose is too high and needs to lower it. The people who are diabetic, they don't make insulin, they can't lower the blood glucose. Insulin is a hormone. So proteins can be hormones. Proteins can function for movement. They're called motor proteins. That's as simple as cilia and flagella on cells to the actin and myosin, the proteins in our muscles that cause our muscles to contract and relax. They can be used for defense, like producing antibodies to help fight off pathogens. They can be used for transport, like hemoglobin. What does it transport? Oxygen. And they can be used as receptors, like in our nerve cells, to know to move your hand. They can be on a cell membrane to receive a signal to tell a cell that it needs to make something. And it can be used for structure, like keratin. Where do we find keratin?
In your hair.
Hair? Fingernails?
It's opposite.
Your hair is protein. Your fingernails, protein. All right?
Isn't it dead protein?
Well, proteins aren't alive. It's made from the cells. Like if you talk about your nail bed or your hair follicles, that's where the cells are that produce your hair and produce the keratin for your nails. But your nails are not alive. Your hair is not alive. It's just proteins that are being protruded out of cells.
I'm definitely not making that.
Now, sometimes proteins are made in a correct shape, but something happens to that shape and they don't work anymore, and we say they are denatured. So denaturation is the loss of a protein's shape. And when its shape changes, can it work anymore? No. So here we have an enzyme with a substrate going to fit in it, and it fits, but then something happens, the enzyme changes shape. Can the substrate fit anymore? No. What are the, what factors can cause denaturation?
Temperature changes.
High temp, pH, what else? Salt, salinity. I think I'm hungry because I kept hearing you say sonic. So high temp, changes in pH, salinity, and they denature by causing disruption to the bonds that give it its shape.
So is your hair like denatured?
No, but if you've ever taken your hair and like, you probably haven't, but like if you put something hot on like a curling iron and you leave it too long, when you take it out, it's like shriveled up in different shapes. That's because you denatured this protein. So, now some proteins don't work not because they were denatured, meaning they worked and then they no longer work because we changed the shape. They were never made the correct way. They were folded up incorrectly. And when they were folded up incorrectly, it alters the protein's shape, which then alters the protein's function, right? Maybe the protein doesn't work at all. Maybe it only functions part of the way. What are some examples of diseases or disorders that are caused by misfolded proteins? Okay, lactose intolerance. That enzyme, you make it in the wrong shape. What else?
Oh, you said frying your hair was one?
No, that's denaturing.
Oh, yeah.
Sickle cell anemia. Hemophilia, no, because you don't make the protein. But, good idea. Mad cow disease. Cystic fibrosis. Parkinson's. Duchenne's muscular dystrophy. There's lots more. But all of these have to do with the fact that we are making a protein that is misfolding, caused by mutations that can cause that misfolding because there might be one amino acid that's different. We are almost done. I'm getting there. You ready? Questions on protein. Let's see how fast. Let's see what we can get through on nucleic acids. Uh-oh, where'd they go? Fill in what. Well, apparently it's not there.
It just doesn't want to do it.
God's giving us a sign here.
Oh, is he?
It's close to that time. You should.
If we do this, you don't do any notes tomorrow. Let's see what we can get through. Let's see what we can fill in. Are you ready? What elements are nucleic acids? Carbon, hydrogen, oxygen, nitrogen, and phosphorus. What functional group is in nucleic acids? Functional group, it has a P, so the phosphate group. What are the bonds between nitrogen bases in DNA? Hydrogen bonds. So the bottom one is hydrogen bonds. The bond between the phosphates and sugars on the side, that's where we get a special name. That name is phosphodiester bonds. So that's the bond where one phosphate attaches to the sugar of the other. Nucleotides have how many parts? 3. What are they? A phosphate, a 5-carbon sugar, and a nitrogen base. Am I telling you anything you didn't already know? Good. There are two types of sugar we can find in nucleotides. What are they? There's ribose and deoxyribose. Look at the picture of ribose and deoxyribose. What's the difference? Bottom right, look at ribose. There's a hydroxyl group, an O and an H. And on the bottom right of deoxyribose, there's just an H. It has an oxygen has been removed. It's deoxified. Not really the right word, but, right? An oxygen was removed. You see that?
Yes.
All right, nitrogen bases. There are 4 bases in DNA. What are the 4 bases in DNA? Adenine, thymine, guanine, and cytosine, right? Remember those? Adenine, thymine, guanine, cytosine. What are the 4 bases in RNA? Adenine,
Yeah.
uracil, guanine, and cytosine. Those are the 4 bases that are in RNA. Now those bases get divided into 2 groups. They're purines and pyrimidines. Look at your pictures here of purines and pyrimidines. What's the difference in the picture? Okay, the overall structure of it, what? 2
Ring.
2 carbon rings. The purines have 2 rings, right? So there's 2 rings. The ones that are purines are adenine and guanine. That's an A and a G. Here's a helpful reminder. Pure, good, angels have 2 halos. Halos are the rings on an angel's head, right? So purines, guanine and adenine, are double rings. The pyrimidines are what's left. They're single rings. What bases are going to be pyrimidines? Cytosine. Which one's in DNA only? Thymine. And in RNA? Uracil. So that's how they're categorized. All right, we will finish these last 2 columns tomorrow. If you are taking your Cornell notes, bring them with you tomorrow. I'm not grading them, just bring them with you, okay? No, you need to bring your book, just bring your Cornell notes. We'll finish these. We have an activity we're gonna do. It's gonna be a good day. Last night of reading, pages 75 through 87.
What about our next year's revision budget?